Today we are going over chapter 3, which are lab tools or tools in laboratory for studying microbiology. Your textbook goes over 6 items. They call them the 6 I's, starting with inoculation, incubation, isolation, inspection, information gathering, and identification. And we'll go through all these methods throughout this chapter. Start off with the microscope. Main components of a microscope are the magnification, and that's simply the ability to make something look larger, and then the resolving power, and this is to show detail on as an image instead of being blurry or out of focus. So simple. Magnification, again, making it look larger. Most people have used a magnifying glass at some point in time. By the time you've been in college, you've probably used a microscope of some kind also. Again, simple magnification is making it look larger than it actually is by bending the light waves. I have a standard microscope, your eyepieces, your nose piece, your objective lens, your stage, your coarse and fine adjustment knobs, your stage, the slide adjustment knobs on there. You have a condenser, a light source, base, the arm. How magnification works is the light comes through, it gets flipped around through the lenses and prisms bouncing around. So the real image is going to be different than what your ocular image is because it's going to be upside down and backwards for your point of view in the microscope, which is why on this type of microscope with the adjustment knobs are much easier to use than the old type with the just stage clips where you just have to use your thumbs to slide them around. Total magnification is the magnifying power of your objective lens plus or times your ocular power, and that gives you your total magnification. Most eyepieces are at 10x, so they magnify 10 times. Your objective lenses usually go 4 for a scanning, so that gives you a total of 40. 10 for a low power, so it gives you 10 times 10 is 100 in total magnification. 40 for your high power, which would give you 40 times 10, so 400 for your total magnification. And then in microbiology, we use what we call oil immersion lenses, which are at the objective lens is at 100 magnification, so 100 times 10 is 1,000 at this point. Again, resolution is giving you detail. If you don't, and they're showing you these circles, the differences between high resolution and low resolution. This high resolution, you can see the overall, low resolution, you can see the overall shape of it because you can't get in to see the fine detail between the fingers. But if you have a better resolution, you can get in and see the actual finger sticking out so you get a better picture of what it is. That's what resolution is. We use oil on the oil immersion lens because at that point the resolution fades a little bit because as you can see on this side of the air, some light leaves. Oil has the same refracting ability as glass does, so it actually captures more of the light, and if you have more light then you're going to have better resolution. So we use oil on our oil immersion lenses to give us better resolution. We can see without the oil on there, but it's a little fuzzy or a little blurry. Magnification, here we go. These are types of bacteria. Here's a yeast cellar, so you're giving you your overall examples of them. There are some that are very small over here. Viruses, unless they are really large viruses, we will not be able to see them. And notice that these over here are not resolvable. We're not going to see a lot of detail on these. These are large enough that we can see some detail on them with light microscopes. So I got to get the best resolution. In this case, we're going to want short wavelengths. The shorter the wavelength, the better the resolution. All right, now types of microscopes that we use. Bright field is the most common type. We use this, we can look at living specimens or stained specimens. If you've ever used a microscope, you've used a bright field microscope. On the opposite of this is a dark field microscope. You don't get direct light going through it, but it does help you with looking at living organisms. We don't use this in our lab. There's a fluorescent microscope. This type of microscope uses ultraviolet radiation. You coat the specimens and they fluoresce based on the types of dyes and types of species they are. But you cannot look at these with the naked eye because the ultraviolet radiation is damaging to the eye. But you can distinguish between different species using this one just by because they fluoresce in different colors. The crown jewel of microscopes is the electron microscope. Instead of using light waves, they use electrons, hence the electron microscope. And these have much shorter wavelength. It's not truly a wavelength, but they're similar to wavelengths, but they're, again, 100,000 times shorter, so we get much, much better resolution on much tinier objects. So these things can magnify up to 1,000,000 times. We can see some really tiny particles. We can see down to protein structures with these. Very similar to a light microscope, but instead of seeing the light, instead of the light waves passing through it, you have electrons, and instead of lenses, you use magnets to focus them, and you get great detail with this. Two types. The first one is a transmission electron microscope. These will pass the electrons through an object, so denser objects will appear darker. So you'll see some spots in there. But these transmission electron microscopes give you a two-dimensional picture of what you're looking at, but you can still see great structure in them. A scanning electron microscope, on the other side, on the other hand, the specimen is coated with a metal, and then you get great 3D images like this bottom one. Up here on this top one, this is the edge of a E. coli, and these are the viruses attached to the surface of the E. coli. You get great detail on these. Again, these things are, you can see really small objects. But the scanning electron is going to be a 3D image, and the transmission electron is going to be a 2D image. Types of slides. Wet mounts and hanging drops. These are for looking at living organisms. Wet mounts, you put water onto, or a specimen onto, usually water, on a slide, and you cover it with a cover slip. You can see the size, watch them moving around. You can see some arrangements on them. Hanging drop is just like it says. Instead of being directly on the slide, you put it on the cover slip and it hangs down into a well on the slide. Most of the times you've probably looked at a fixed smear. These are already pre-stained and pre-preserved. These ones last a long time. You can see lots of things on with these. Types of stains now that we need to go over. Our basic dyes are going to have a positive charge on them, and they will attach to the negative charge, components of the organism you're looking at. These will give you a positive stain. A positive stain means that it's staining the specimen. So you see the specimen absorbs the color and it appears at a different color, whatever color you happen to be using. And the background is going to stay clear or white. On the other side, we have acidic stains, which have a negative charge and will stick to, again, because opposites attract, will attach to positive charge elements in the organism. These can give you a positive stain. In other words, again, the specimen itself is what's stained, where you see the specimen colored, and that's what's showing you here. Then there's another type of staining we call negative staining. And this is where, again, the opposites, like charges will repel each other. So you have it's a negative stain, and they'll not bind with the negative components of the cell. So what happens is the background is stained and you see a blank space where the specimen is. So that's a negative stain. Types of staining: simple stains where you're just using one dye, so you like methylene blue, or carbol fusion, malachite green, crystal violet, and you'll end up with just one color. Differential stains use two separate stains. You'll use a primary stain and a counter stain. Gram stain is the most common one for this here. It uses crystal violet as the primary stain. Then there's a few other things involved in it, but you'll have safranin counter stain as your counter stain. And this will distinguish between purple and red colors depending upon whether something is gram positive or gram negative. Structural stains. These are stains that will identify specific structures in a stain. A capsule stain does this. Flagella stain, so you're just looking at the structures, not necessarily the cell. The cells will absorb some color too, but you're doing it to just try to identify the structure, whether it's present or not. And then whether, if it is present, what type of arrangement it's got, and for the in the case of flagella. These are examples. Simple stains is using methylene blue, so it's a blue stain. This is a negative stain, so the specimen, you see the squiggly line, is not stained, but the background is. These are differential. These are gram stains. Over here is a gram positive. Here's a gram negative because you're using two different colors. An acid-fast stain, you got two colors in there to distinguish between some characteristics. Spore stains, the cells are red. The spores are going to be green in this particular one. Structural stain, you have the capsule. It's similar to a negative stain. You have that space around the cell, and the background is stained quite a bit. Here's flagella stain, the flagella. This one here is a fluorescent stain, and it's looking for the cell membrane, and it's for that particular structure on it. All right, again, our 6 I's of culturing. Inoculation is the first thing, and this is where you're taking a specimen, you're putting it onto a culture, you're trying to grow a culture of it so you get to see observable growth. And this is, you're putting it onto a particular media to get it to grow. Then you have to isolate this. Most of the time when you're going to be growing something, unless you have it already isolated, you're going to have a mixed culture. Then you want to separate out the various types of colonies and growths going on. So that's where you're getting the isolation. And then you grow this under certain conditions. Some organisms like cooler temperatures, some like warmer temperatures, some like lots of oxygen, some don't want any oxygen, some want higher carbon dioxide. Whatever the conditions, you'll incubate it under those conditions to grow them. Then you get into the inspection, information, and identification, which is coming up. So again, that isolation, you have your specimens, you grow them, and they will look different. Usually you can see, not always, but you can see differences in colony, whether it's the shape of the colony, the size of the colony, the color of the colony. And then you want to isolate that out so that you end up with just one type. Isolation methods. The most common ones that people use are streak plate, where you'll take a specimen and you'll spread it around in different areas, and it'll end up looking like this here, where you have a mass, and then it gets a little bit thinner, so that you end up with isolated colonies. And in this case, you can see 3 different types of colonies: a yellow one, a red one, and a green one. Excuse me, a clear white one. You can do pour plates, where you pour them on 2 plates, and then you have them random all over the place. If it's, in this case, it's most likely going to be in the auger also, but again, you can see individual colonies spread around. And then we have spread plate, where you put your sample onto your auger and you spread it around using different tools. So you have it all over the place, but again, you can still see differences in the colonies. They're giving you examples of color, but you can always tell by the different sizes. Again, these red ones are much larger colonies compared to these yellow ones. You can see them up here also. So look for size, shape, and color. you need to inspect it. Did you have a pure culture? A pure culture is one where there's only one type of species, like this yellow one right here in the middle. There's only one species in this. A mixed culture is where you've got two. So over here you have the yellow and there's white ones in here too. And they're kind of spread throughout. So that was a mixed culture. This middle one right here looks like it's a contaminant because you've got the red all around it and you get this one patch of the white in here. But notice again, the shape of the colony is different, so you can see these, let alone the color and the size. This one here is most likely contaminated because it's just isolated here. Over here, you have the white and the yellow all over the place in it. So that one there was a mixed culture. And over here, they're showing you pure cultures. There's just that white culture, the yellow culture, and the red culture, individual ones. How do we identify these things? Again, we look at this individual cell. So we'll look at the shapes of the cells, how they're arranged. We look at the colony, look at the size of the colony, the shape of the colony, the color of the colony. In modern times now, we can use DNA sequences through a PCR method where we make lots of copies of the DNA, then we read it. We can do biochemical assays where we test for different chemical reactions. And that's kind of what they're showing you right here at this flow chart here. They're testing to see it. They see that it's a gram-negative coccus and coccus bacillus. So then they test to see if it has the enzyme oxidase, so it's negative or positive. And if it does not, then it's probably this thing. If it does, then we got to do a few more tests. Is it fermenting maltose? And that's what we're doing, biochemical tests there. And then there's immunological tests. Immunological tests test to see if it reacts to various antibodies that you produce, or various other chemicals we can put into it. The media itself, we have liquid. There are three types: liquid, semi-solid, and solid. Most of the time, you're probably just going to be dealing with liquid or solid, but there is a semi-solid. Now, the composition of them, you have synthetic, and this means we know exactly what is in it and exactly how much is in it of something. Complex is, we're not 100% sure about something. An example of, well, we'll get to that in a moment. Then we have different types of agar. You have a general purpose, which is used to grow lots of different bacteria. You have enriched. Enriched agar is meant for growing bacteria that are picky, that need something special in it. Selective blocks some things from growing and enhances others growing. Differential will show a difference between some of them. Anaerobic is without oxygen. Transport ones are to see if they can move around. You can do chemical assays, or you can do enumerations where you're counting. Again, liquid one is a broth that does not solidify. Semi-solid does contain a solidifying agent, but we don't use those very often. The broth and the solids are the ones that we use the most. A solid, again, you can heat it up and it'll form a solid surface here, and you can do different inoculations of this. And here's a stab. You can see the bacteria growing in the middle of it. It did not grow, move out very far, so this is probably not a motile one in this particular case. In this case, you see that it's very cloudy, so this one here can move. So this one's got flagella. This one turned black, so that means it's going to produce some hydrogen sulfide. So there are some characteristics we can tell, or some characters we can tell just by using various types of media for it. Agar, it's derived from seaweed. It's a solidifying agent. You mix it up, you heat it, it turns into a liquid, and then you can make it into whatever shape you want. In a tube, you can make a slant. You see how it's kind of slanted here. You pour it into a plate, make a plate, but it can be remelted and to change the shape of it. The agar itself, most organisms do not digest that particular protein that's in the agar, or that solidifying agent. So the organisms will just grow on the using the nutrients that you provide in your media. Some common ones are nutrient broth. This is containing a beef extract and peptone, There's no agar in it, so it's not. This is going to be a liquid. Then you add the agar to it, and it's the same thing, and it becomes a nutrient agar. Again, this is a general media. We use a lot of tryptic soy in our labs also. Synthetic, again, contains, it's pure organic, and we know the exact formula. A complex or non-synthetic is where it's got something we're not 100% sure what it is. So I'm going to go back one. So that beef extract. So when we take something from an organism, from another organism to put into it as a nutrient, we don't always know the exact makeup of everything. So we can take blood and make blood agar. That would be a synthetic, a non-synthetic one. That would be complex because we don't always know the exact proteins and everything involved in that blood. We're pretty close, but we don't know exactly everything. Again, general purpose medias, they grow a broad range of microbes, and they're usually non-synthetic because we're putting something into them. Enriched, these are either blood serum, hemoglobin, or something special to grow a fastidious. I want you to know that term, fastidious microbes. Fastidious means that they are very picky. They're hard to grow unless you have these enriched media. So know that term, fastidious. Here's some examples. This is blood agar, this red color here, and note you can see some clear halos around some organisms and not defined halos around others, and that gives you a little bit of a chemical analysis. Also tells you a little bit about what types of enzymes they have in there. But this is one that helps grow. Over here, this is a chocolate agar. No, it does not have chocolate in it. It's kind of a cooked blood agar, and it's used to grow various microbes. Both of these are for fastidious organisms. Selective media is going to contain something that is going to inhibit the growth of an organism. A differential medium is going to allow organisms to grow, but it will give you a visible difference. So they're showing you on here how you have the purple and the green. So it'll show you a difference between the organisms. And right here, this is a general purpose. You get lots of different colors, sizes, and shapes of bacteria. On the selective, they're only showing you the one color, this red, because it's inhibiting everything else. And on the differential, it's showing you the difference between two by showing you a color change. Sometimes a medium can be selective and differential at the same time. This case here, this is going to be the chromagar. It's showing you the different species grow with a different color. Since you're seeing a visual difference in them, this one is a differential medium. So chromagar is different, a differential. This one on the left is mannitol salt. It is a selective media made to grow Staphylococcus. And then it is, dang it, it is also a differential media because, whoa, go back to where I was. What happened here? Because the pathogenic Staphylococcus will appear yellow and turn the background yellow. So it does differentiate a little bit. Over here we have McConkey agar. McConkey is selective in that it only lets gram-negative bacteria grow, and then it's differential because the ones that have the red these are bacteria that can ferment lactose. Down here in these tubes, you have triple sugar iron agar. So they have 3 different types of sugars plus an iron in it. This first tube has no growth in it. This second tube has some growth, but there's no acid production. And the third one here, you see some acid production here at what we call the butt of the tube. It's changing color. This fourth one has is a bacteria that grew and had acid production throughout the tube. That's why it's yellow. On this fifth one, again, you had a bacteria grow, kind of sit in the middle here. You had acid production in the butt, so it lowered the pH. And notice this black region here. That's because the bacteria are reacting with the iron and producing hydrogen sulfide. And again, here's that chromogar. You've seen that one. Another media to show fermentation, a lot of times you'll put a tube into it, and if it ferments, it'll produce carbon dioxide, and this is where that little gas bubble is. And if it produces acid, you'll have a pH indicator, because it's normally red, and if it produces acid, it'll turn yellow. So this particular bacteria, you can see it down here at the bottom, produces acid and gas, as you can see the bubble in it. And that's all for this one. Have a nice day.